Hi everybody, I'm Dhanalakshmi. Today we're going to discuss about photosynthesis in higher plants. First, we have to know what is photosynthesis. Photo means light, synthesis means preparation of food. Plants are autotrophic organisms that can able to sense own food. Likewise, cyanobacteria also undergoes the photosynthesis process. The plants get energy from sun by converting solar or radiant energy into chemical energy, process called photosynthesis. In simple definition, it is a physico-chemical process by which they use light energy to drive the sense of organic compounds. That is, light energy is converted into chemical energy. This is explained by the simple equation that is given below. 6 CO2 molecules which is comes from the atmosphere, water which comes from the soil, absorbed by the roots. These two inorganic compounds involves in synthesis of organic compound called glucose molecules. So this is the organic compound. This is the main product. Two other byproducts are water molecules and O2. The O2 evolution is taking place in the process called photosynthesis. Okay, students, first we have to know how does the different scientists explain about this process. Number one, here Joseph Brisley, the scientist, first time studied about the plant growth need air. He is the person who discovered the oxygen. Plants restore to the air whatever breathing animals and burning candles remove. And second side is Jan Engine House. He studied about only the green part of the plants, that is leaf or young stem would release oxygen, that is inhale. Bubbles are the indication of oxygen evolution in the plant green parts. And third, Julian Van Sachs first studied about the glucose. It's made in the green part of the plant, that is special body called chloroplast, which present in the green part of the plant, usually stored as starch. So the glucose is stored in the form of starch that is studied by the Julian Van Sachs. And the T. Engelmann, he studied bacteria accumulated mainly in the region of blue and red light split of spectrum, that is wavelength. The different wavelengths are responsible for the observing the pigments. Blue and red is the very important wavelength which undergo the photosynthesis process. And Collins Vanning, who explained photo is essentially a light dependent reaction. So it is a light dependent reaction. It's explained by the Collins Vanning, in which hydrogen from a suitable oxidizable compound, maybe it is the H2S or water molecules or the oxidizable compound that reduces the CO2 into C6H12O6. So H2S or H2O, from that one the hydrogen which reduce the CO2 into glucose molecules. This is explained by the simple flow chart. You can see the flow chart here. See, photosynthesis is a two process. One is light reaction and is a dark reaction. Light reaction is a light dependent reaction. It occurs in the presence of light. And dark reaction which occurs in the absence of sunlight, means it is occur in the daytime, but it does not need the energy. That is what you have to remember. Dark reaction means it's not, you never understand it is occurring in the night. That is wrong. Dark reaction also taking place in the daytime. Very important. Understand, student? It occurs in daytime, but in the absence of sunlight. But this dark reaction use the light energy. So light reaction produces two important energies, ATP and NADPH, and O2 is the byproduct. Light reaction taking place in the thylakoid. The two process called cyclic process and non-cyclic process. Both of the process produce the ATP and NADPH and evolution of O2. This is the light reaction product, whereas in the dark reaction, CO2 is reduced and produce the glucose molecules by using the ATP and NADPH. Now I explain this one by using simple diagram for you. So first we have to know where is the this process taking place. Right? Just drawing for you the diagram, the leaf structure is a lamina, a flattened leaf-like structure is a lamina midrib, and then veins and veins. Just cut in the small portion of this one, and this is the region. I will show the TS of the leaf structure. This is the upper epidermis, below the lower epidermis. So upper epidermis, lower epidermis, which is very important for the production of internal vascular tissue and palisade and spongy pad. Now the below the upper epidermis, the parallelly arranged compactly elongated structure of palisade parenchyma cells arranged in 2 or 3 layers form. Now below the palisade parenchyma, there is a irregular arrangement of spongy parenchyma that I will draw it for you. First I will complete the palisade parenchyma, then I will just draw the spongy parenchyma. So now you can see here spongy parenchyma is arranged loosely. It is arranged in loosely manner. So all the part it's palisade. Below the palisade parenchyma, spongy parenchyma is arranged like this. So students, it is very important you have to remember this one. And simple thing, you must understand below epidermis region what is present in a God cells. So exam point of view, they will ask you what is God cells. God cells are modified lower epidermal cells. So these are the two adjacent God cells. Two adjacent God cells. What is God cells? God cells are modified epidermal cell, lower epidermal cells, which involves in the exchange of gases. So this is the pore, a small pore which is regulated by the two adjacent God cells. It's called stromatal pore. So students, you have to remember stromata or stromata, it's nothing, it's a pore which is regulated by the two adjacent God cells. Now I just want to show you the pathway of the pathway of exchange of gaseous, that is release of water vapor as well as the exchange of CO2 from the atmosphere into the leaf. So now I just show you the movement of the CO2. Now the CO2 enter into the leaf and it is will pass the interstellar, these are the interstellar spaces, and finally reach in the palisade parenchyma. So now the CO2 is moved into the palisade parenchyma cytoplasm region. Now I show you the single parenchyma, palisade parenchyma cells. So this is the single palisade parenchyma cell. Just want to show you only one single palisade parenchyma cell, and that is regulated by the cell wall. So it is the cell wall. Within the cell wall, the plasma membrane layer is present. So just want to draw the diagram. This is the plasma membrane. The central part is occupied by the large central vacuole. So students, please remember vacuole is irregular shape. In the periphery region, what is present? The nucleus is present. I just want to draw the chloroplast. A single chloroplast, more than 20 or 40 chloroplasts also present. So these are the cytoplasm. The cytoplasm fluid matrix and mitochondria is also present. Mitochondria, and then here endoplasmic reticulum, everything is present. I just want to draw this single chloroplast structure, then you can understand very easily. So this is the single chloroplast. It's a double membrane organelle. This is also they'll ask the exam point of view. What is chloroplast? Photosynthetic organelle, which is covered by the double membrane. This double membrane organelle, which is consist of the two compartments, thylakoid, another one is stroma region. So what is thylakoid? Stack-like arrangement of the single coin is thylakoid. The group of thylakoid, collectively called as gram. So this is one single granum. Likewise, more number of grana is present, then it is called grana. In plural, it is called what? Grana. So one single stack like structure coin which is called thylakoid. Group of thyla collectively called as grana. So like if one, two, three, more number of grana is present, then it is what? Grana. In plural, it is called grana. And exactly the light reaction taking place in the thylakoid. This is the fluid matrix region. In this matrix, it is only what? Carbon fixation is taking place, and this region is called what? Stroma. So this is the stroma region, and here what is present now? Very important. It is that the chloroplast is DNA, RNA, ribosomes, enzymes, proteins, and lipid molecules. So you have to remember it is a cell within a cell called as chloroplast because it contains its own DNA, so it can able to synthesize its own proteins for so the protein can able to act as a enzymes to follow the carbon fixation process because carbon fixation is the enzymatic process. Now you can understand very important structure of the chloroplast process. So I hope you understand both this demo class. Now rest of that means the rest of the questions we will discuss in the later classes. Thank you so much.